Hey everyone, Kim here today. I wanted to talk to you today about the methods we use to quantify wildlife. And let's take a moment to look at the learning objectives for this particular lecture. When we're talking about quantifying and measuring wildlife, the first thing we want to talk about is the importance of planning for this. We need to think about our question, we need to think about our wildlife of interest, and plan out our approach. We're also going to discuss measurements related to populations and population management, but we'll also delve into measurements related to habitat and habitat measurement. So let's start at the beginning and discuss why it's important to quantify wildlife. There are three categories under which knowing the number of animals is pretty helpful, and the first is to assess environmental conditions. If we know a bit about how many animals are or are not in the area and then can relate that to an environmental characteristic, it ultimately allows us to create better management. So, for example, we can look at a study of black bears in New England. Researchers wanted to know how the probability of occurrence of black bears was influenced by the amount of development in nearby the site where they were looking for these black bears. And to answer the question, they not only looked at the number of black bears in the area, but they tried to look across areas of high, medium, and low levels of land development. What researchers ended up finding is that as the amount of development within 5 kilometers of the sites increased, there was actually a lower probability of occurrence of black bears. And you can see that in this graph below. And in this case, it actually related directly to the landscape conditions. Black bears were really just looking for suitable habitat, and so as development increased, there was less and less suitable habitat for that black bear population to be able to hunker down in. It relates directly to the management of this area as well, with positives and negatives here. Because if we're trying to support black bears in the area by lessening development or by allowing for more suitable habitat in areas of development, we could see potential conflicts arise between humans and bears. There would likely be more human-bear interaction if that development or if that suitable habitat was protected in the area. So you can see that even though we now have that information based on the environmental conditions, how we use it is going to be a bit tricky. Now, the next piece that is important to consider when we're looking at quantifying wildlife, or the next reason I'd say, is so that we can understand how many animals may be harvested if that's of interest to management. And when we think about that, by estimating the population, we can then have the ability to model and understand the surplus or the quote-unquote extra that might be available for harvesting. And then finally, by understanding the specific relationships with these environmental conditions that we were talking about earlier, we can also look at the potential impact of environmental change on those populations. And in particular, we can understand a population's resiliency. So let's think about this black bear study again. If we're in a situation where development continues to increase and there are more and more land that's being developed within that 5 kilometers surrounding sites where black bears are located, we can see and project that our probabilities for black bears to occur will become smaller and smaller within this region. And so these are the ways that we want to use this data. And to do that, we have to think about the best ways to collect that data that answer the specific questions. So what is it that we're quantifying? What do we want to know about these areas? Now, there are a couple different population numbers that we can think about when quantifying wildlife. The first is abundance, which is just the number that you find in a particular count, just the overall number. Whereas density adds a piece of geographic limitation to that. So density is the number per spatial area. And you can think of this as number per square mile. You can think of this even if it's much smaller than that. And then also we can talk through some demographic features as well, like births and deaths. We can think about immigration and emigration. Now, when thinking about population numbers, we can also look at habitat use to get at that number as well. So a couple of the different pieces of habitat use that we can discuss, and I'll give you an example of one in just a moment, is occupancy, meaning are they even there? Or are they somewhere else? There's selection, meaning are they choosing to live in one area versus another? Like in our black bear example, bears were choosing not to live in areas that were heavily developed. And then also, what about their behaviors? How are animals moving between areas, and what does that indicate about the habitat? So, for instance, each fall at Hawk Mountain Sanctuary, which is in eastern Pennsylvania, interns and wildlife professionals will sit all along ridges in the area to complete a raptor count during the annual raptor migration. And so these workers are taught to ID birds of prey both by sight and sound so that they can count how many individuals are passing through and which species each individual belongs to. So you can see this is quite successful in terms of being able to attract these birds and being able to get a count on or an idea of what overall number there are. But there are a lot of other ways to do this as well. You don't actually have to see the organism in order to get some of these habitat use numbers. You can also do this acoustically, and we do this a lot when we're talking about bats and birds and frogs. But because there are so many ways to go about doing this, getting abundance numbers and also understanding habitat use, what's really important is that we know what question we're asking in order to know which level of quantification we need. Ultimately, we have to know a little bit about the wildlife of interest and what information we want to collect in order to decide how exactly we want to count them, whether we get abundance or density, whether we get birth or death, etc. So let's think about some general guidelines to the field methods when we're thinking about wildlife. The first and probably most important is, what's your question? What exactly are you asking? Because that will help you determine what it is you need to be looking at. And that leads us into the second and third points, which are actually relatively related, but they're each important. So the second is we want to collect the data that are specific to our question, which is point number 2, but that also means we don't have to collect everything. Not everything that we can measure should be. We often see this occurring when someone says, like, they're interested in bird behavior, for instance, and they want to see how bird behavior changes, like around particular vegetation, for instance. So they could measure just about everything possible, thinking air temperature, soil characteristics. They could look at bird size and type. They could look at vegetation size and type. But the most appropriate way to go about doing this is to collect only the data needed to answer the question at hand. This also leads to our fourth piece, which is whether or not you want to answer a question or push an entirely new idea. And this can become a difficult thing to move through when you're trying to come up with novel research. So it's really a balance routine. So for instance, it could be a matter of very applied science in which you're trying to simply learn more about a species, or are you interested in developing a new idea of predicting maybe how a species will respond to other changes? It's important to try and delineate that before setting forward on your method. The fifth component is thinking about what is needed in order to answer these questions. And that's because when we start thinking about sample size and how to address any sort of error that we may have in measuring a species, we're better prepared to take those measurements. And the last component, the importance of thinking about the biological significance of our conclusions. And this is always tricky for people. Just because there is a statistical significance with something does not necessarily mean that it's going to make any biological sense. And this is certainly true when we get into the ages of big data, in that we can have enormous quantities of data, and it becomes very easy for those quantities to automatically have a significant statistical result. But that doesn't necessarily mean there's a biological reason for that. So when we're talking about studying populations, let's think about how this population data may be collected. And there are 3 distinct ways that you can think about collecting population information. There's a census, there are population estimates, and there are population indices. Now, a census, which some of you may be familiar with because it's conducted throughout the United States every 10 years, a census is a complete count of a population. In practice, this is actually very difficult to do and really only occurs when we have endangered species or a tremendous amount of resources and effort, or in some organisms that have given behavioral patterns in which all the individuals can be counted. So for instance, an island species would fall into this category. And also thinking about seabirds that come to a specific location to breed. If they have a behavior that makes it so that they come to one area, then you can potentially get a census. But overall, this is a very difficult measurement to gather. So more often than not, we go after population estimates. A population estimate is where we set some sort of time and or spatial boundary, and then we get our best count of a subset within those bounds. And we use that to estimate a larger population. So for instance, we could use an example like the MARC recapture study approach. In a particular area, we could capture as many squirrels, let's say, as possible and tag them with a harmless lasting marker, making note of how many individuals we were able to do this with, how many individuals we collected and marked. We could then come back to that area after releasing those tagged individuals and repeat the exact same process, where we try to capture as many individuals as we possibly can. So, comparing these, if we have, after that second capture, if we have a number of them with the tags that we originally placed, we know we probably caught most of the individuals in the population the first time around, and that our captured number is close to that overall population number. But if we try to capture these squirrels the second time and realize that only a small percentage of them have tags, we know that we're likely missing a whole bunch of squirrels in the area. And there are equations for how to get at these numbers, but we're just sticking with this as an example for now. And as you might imagine, there are a lot of ways this number can be impacted, so it is just an estimate, as we said before. We know we can't actually capture every individual in the population, and that we'll have to use some way of extrapolating the numbers we did catch in order to get a sense or an estimate of that overall population. So we know there's some sort of error involved, which is the biggest difference between an estimate and a census. That brings us to an index, a population index or an index of relative abundance. So if we take an estimate multiple years in a row, we start to develop an index. An index is a long-running estimation of the population. So the National Breeding Bird Survey is a prime example of the power of indices and the important data that they can provide towards monitoring and conserving wildlife. In this case, the Breeding Bird Survey is conducted annually since 1966, and it's driven by volunteers collecting data along a variety of bird breeding survey routes, which those routes are distributed all across the country and into Canada. And these have been invaluable in terms of documenting a distribution of species like the scarlet tanager. And with all this data, we can actually look at trends of populations over time. So for instance, the northern bobwhite, we begin to see red starting in about 2013. So this census has been collected from 1966 to 2015, but starting in about 2013, we see this big patch of red. And what this is telling us is that we're seeing a large decline in our northern bobwhites over time. And so this can flag us to know that this population in particular deserves our attention when it comes to management, because we're seeing a big shift in their population. So we need to understand why and really hone in on what it is we can do to potentially protect this population from declining any further. But when it comes to estimating a population, as you might imagine, we know there's going to be error. So let's talk about what that error looks like. It's really important that we quantify and evaluate the error, because we need to better understand what it is so that we can know what types of conclusions we can draw from the data that we collect. So there are three basic statistics pieces that we are going to discuss when it comes to error in estimating a population: accuracy, precision, and bias. Now, accuracy we can probably consider a review. Accuracy is how close the estimate is to the true value. So you can see we have one dot that has fallen directly into the true value, so that one is highly accurate. We have another one that has fallen just outside of the true value, so that one's a little less accurate. And then we have this guy way over here that has no idea what's going on, and so clearly that's not accurate at all. And the next piece would be precision. And this is always the piece that I know I personally trip over pretty regularly. Precision and accuracy are two different things. Precision is how repeatable the estimate is. So here we can see that there's kind of a clumping of our estimations. That means that we have a precise estimate, but it is clearly inaccurate because it's not within the true value. So clumping means precise, but it doesn't necessarily mean accurate. So let me give you an example of something that is imprecise. We have this kind of all around the board type of estimate, and so that would be something called imprecise. It's also inaccurate because we're seeing that nothing is falling into this true value here. And then we can talk about bias. So bias is kind of how precision and accuracy relate together. And our goal is to obviously avoid bias. Bias is a systematic error, or an error that has a pattern to it. So if you look at these panels, 2 of them are precise and 1 is not. However, that precision is clearly a pattern that is nowhere close to the real number. So you can see in this second circle, we have a relatively precise estimate, but it's way outside of our real value. And same thing on the third panel. We have a precise measurement, but it's way outside of our real value. And because of that, we know something is wrong. Something is causing us to arrive at the wrong conclusion time and time again. And instead, if we were a bit more all over the board, but at least surrounding the correct answer, we know we have an unbiased approach. There's no pattern of error, no pattern of how we're arriving at these values that can be explained. There's no cycle with it. So for an example, to give you an idea on this, let's say we were trying to count the number of deer in an area. So we lay out corn in an area to attract them, and we count any deer that will take that bait. But it just so happens that we picked a site that's next to a really loud highway. In our study, we are only counting those that are brave enough to come to the area and get the corn and have a preference for corn as food. But what about any skittish deer that weren't brave enough to go through the loud noise or aren't necessarily attracted to the corn? So how do we account for these individuals? So you can look at that as one of these last 2 circles where we're clearly getting some dominant or aggressive deer, and we're missing a large portion of the population that were too afraid to come into the area with the loud noise. So with that, I want to leave you with this particular table because I think it sums it up nicely. You can see bias in the top 2 panels, with 1 being precise because they're all clumped together. So you see that clumping pattern that I was talking about earlier. And then 1 being imprecise because of the spread between the results. These are not necessarily clumped. They're more spread out, but they're nowhere close to that real measurement. They're in the same general pattern away from that real measurement, and so there's clearly something that is causing our measurement to move in a systematic direction away from the real value. Whereas the bottom panel shows us unbiased measurements. Our estimates are evenly spaced around the correct answer, meaning that our error is not some sort of pattern that we have fallen into. So ultimately, this balance of being able to repeatedly measure individuals is really key to population methods and monitoring.